From Microbe TV, this is Office Hours for Wednesday, April 2nd, 2025. I'm Vincent Racaniello, and welcome to your viral corner of the internet. Welcome, everyone. Good to see you all. Tell us where you're from. We'll shout you out. It's the day after April Fool's Day. Do you know why it's called that? Well, many years ago, a pope, one of the popes, I forgot who, switched from the Justinian to the Gregorian calendar, which was more accurate to the planets and all that. That's what we're using today. In the Justinian calendar, New Year's Day was April 1st. And in the new Gregorian calendar, it was January 1st. So he switched New Year's Day to January 1st. But a lot of people didn't get the news, or they didn't like it. And so they continued to celebrate New Year's on April 1. And they were called April Fools. There you go. Who do we have moderating tonight? Big crowd. Bless is here. Steph is here. Vanity Nutrition is here. Tom Steinberg is here. There you go. Almost complete Julian calendar. Yeah, Julian, sorry. Thank you, mods, for moderating. Thanks, everyone, for coming. What's the plan tonight? Okay. I've had queued up the last 3 weeks. More art of Dixon's. So I'm going to show those. We'll chat until 9. I'll do a mini lecture. And then we'll wrap it up with some more questions. Steve is in Kauai. Welcome back. Rita is in the beautiful Great Basin in Idaho. Considerable pluviosity. I guess you like that word, huh? I did too. Wayne is in Urbana. 69F. What is it here? You know, it suddenly got cold again. Did I bring my phone? Yeah, I did. It's in my pocket. So it was nice and balmy last week. Today was freezing. Bloody freezing on campus. What's the temperature right now? 6 degrees Celsius. So campus is kind of a mess. They closed all the gates except one. Now you have to show your ID to get through it. So the campus is closed off. I don't know why. Why do they have to close off the campus? For me, I have to walk twice as long to get to class. And I have difficulties with that. And then coming back, I have to get on a train to come here and do this, and it makes it a little more tense. So thank you, President Trump, for screwing everything up. Kang is in Chicago, 14C. More rain on the way. Pete is in London. Hello, Pete. Rima, hello from Iowa. Long time away. Welcome back. Here you go from Iowa. Noir LeBlanc from New Mexico. Welcome. Haven't seen you in a while. Nice to see you. Will is in Changsha. It's 12C. And sunny. It's just early morning there, you know. Chris and Lisa from Montreal, welcome. Truth or Dare is in La Jolla. Pat is in LA, we're at 62F. Brian is in Montreal, 1 degree C. Maureen is in Northeast Ohio. Mark Martin is in Tacoma, 10 degrees C. Yes, Noir is in Santa Fe, lovely town. 6,775 feet altitude. Pat is in Nova Scotia. Betty is in Washington Heights. Marius is from Romania, but currently in Montreal doing PhD. Welcome. Angry Penguin is north of Toronto. Jeff Bliss is in Elgin, Illinois, 20C. Hans is in Ecuador, 30C. Now that's warm. That's very warm. Spamlet says it's the day after All Fool's Day here in Bedfordshire, UK. In Iowa, it's 10 degrees. Laura's in Washington, PA, 20C. Hugger, 51 in Watertown, Wisconsin. Double rainbow evening. Hello, John from Twin Cities, 33 degrees C. Eliza is in southern Ontario. Lots of pluviosity, indeed. Lori is in Charlottesville, 13C. Mark says, yes, but I have senior class students. They appreciate your efforts. No, I don't think so. It's fine, though. I don't care. I just teach, you know. They don't have any idea of what I have to do to get there and get back. But that's fine. They don't need to. Hello, Michael from Mexico City, 26C. Carol, our favorite nephrologist, is in San Diego, where she did her training. 16C. SRR, thank you for your support of science communication. Donating via YouTube to raise the algorithm. Okay, let's do it. Jill is from Wilmington, Drizzly, and 47F. AMG, hello. Katya is in Detroit, 8 degrees C. Emily is in Ottawa, Canada, -2 and snowing. Joseph is in southwestern Ohio, Ontario. Hello, Costello from Ohio. Welcome back. Mike and Lori are from Seattle, 51F. Ergotic, Den Haag, Netherlands, 17C. Martha's in Charleston, where it is 22C. AMJ is in Austin, Texas, 82F, which is 28C. Yeah, you can just reverse that one. Markle is on the east end of Long Island, 4C and cloudy. Aubrey is in Richmond, Virginia. Well, everyone, it's another crappy week here in the U.S. of messing everything up, breaking things, and breaking what was otherwise a good product, a good country. We had more firings in science, NIH and CDC, many, many thousands of people fired. The administration is taking its shots at Ivy League colleges, right? Which I have mixed feelings about this. Let me take this off here and I will explain. I understand that the presidents of these colleges need to get the grant money for their faculty, okay? I don't agree with giving away everything to get money. You're giving away academic freedom, then there's no more university. So find some middle ground. You're smart. Figure it out. Don't say you can't do it. Columbia, Penn, Hopkins, Harvard, Princeton. Yeah, the Ivies. Here's Peter from Boston, where it's 276 Kelvin. Keith is in Calgary, 9C. Hello, Veronika Privyet. In Moscow, it's +6C. Ticks ahoy, huh? I'm ready to email the institute that made Sputnik, that being the COVID vaccine. to see if they want to make a Lyme vaccine. I'm sure Mark Martin could weigh in on a Lyme vaccine. EHS Talent is in Lehigh Valley, PA. It still says Q&A in my calendar. That's okay. That was the original of this series. Hello, Joseph, in Stamford, Connecticut. Good to see you. Joseph is a relative of mine. Tractor Poodles in Bangkok. Earthquakes, right? Everything okay with you? I saw some pretty scary videos. Hello, Jessica from Toledo, Ohio. Walter's in north central Massachusetts. 3C outside. TG says one building in Upper Manhattan that Columbia owns would easily get $400 million. They capitulate for nothing. Yeah, well, you know, the president stepped down last week, and now we have a new acting president. I just don't agree with bending over backwards for all their demands. I think you have to have some backbone, and the faculty need to realize it. Money, Lillian says money is not the point of science. Money should be a tool of science. 27C in Bangkok. Tractors, okay, good. I'm glad to hear that. Polka Dot Pup is in North Texas, 74F, waiting for storms. Anyway, I'm going to go back to the top. We had some questions that I skipped over. Les points out that Michael Minna has an opinion piece in the Times. He hates the HHS director's anti-vaccine stance. Of course, there's no reason for it. There's zero reason to be anti-vaccine. There is zero reason to do another study on vaccines and autism. He says that no vaccine is effective. This is a total lie. Everything he says is misinformation and lies. How can this person be heading HHS? Congress, Senate, shame on you for letting this guy lie in your face and then vote yes. I don't care what's at stake. You know what? I don't care if your job is at stake. You can get another job. It's not that lucrative to be a senator. Oh, it's the power you like, huh? Well, tough. If it were up to me, you'd have single terms for everybody across the board, because then it gets rid of some of this garbage. How can you confirm a person who lies and spews misinformation constantly? And, you know, Peter Marks, head of CBER at FDA, that's the division that takes care of vaccines. He resigned and said, I can't deal with the lies and misinformation. Good for him for speaking out. But you know, it's only going to get worse. They're going to have a head who's a fringe anti-vaxxer. And yeah, fringe meaning that you don't do anything good and you just lie. They're going to repopulate the committees probably. Horrible. Horrible, horrible, horrible. So Kang says, could we have a series of mini lectures on the microbiologists who developed the germ theory to create a pluviosity of truth to wash away the lies of the kakistocrat RFK Jr.? Yeah, I'd be willing to do that. It would take some time, right? But I'm willing to do that. Maybe some of my colleagues could help. Germ theory. You know, it's clearly been proved. Miasma has been disproven a long time ago. And if he believes that, then he's got some issue because he doesn't know how to interpret data. Kakistocrat. Ay. Kang says the mini lectures, perhaps from Michael Schmidt, could be made into shorts to inform even the least educated voter to become microbiologically literate. Maybe even Mark Martin would help. Mark Martin, Michael Schmidt, me. We could just do short mini lectures. Yep. Hello, Tom. I forgot to point out where you are. Oregon Coast Range. Good to see you. Pete said, I thought about Dixon and his love of jazz on Monday. I went to the London Jazz Cafe to a 55-year celebration of Miles Davis' album, Bitches Brew release. Doubt Dixon's period for Miles, though. He didn't really mention it. He just talked about something blue, right? Hmm. Okay, Will asks a very good question, and I will tell you ahead of time. I don't know the answer, and nobody knows the answer. But we talk about all this all the time in my course, and I showed you a table last time of memory, durability of memory after infection with viruses with mucosal reproduction versus systemic reproduction. So Will's question is, many systemic diseases give immunity that remains effective for years, yet many respiratory diseases have immunity that lasts a few months. Why? So let's pick up the lecture where I mentioned that. I don't know where it would have been. I mentioned it last time. So that would be adaptive immunity. So let's go there. I brought it up last time. I want to show you again the data. What is adaptive immunity? 10. Nope, that's assembly. Then we have that. Then we have innate, blah. It's either 13 or 14. Sorry, folks. You can just chill while I'm looking for this. Yeah, it's lecture 14. Adaptive immunity is associated with memory. Now, here it is. The disease in the left column and the persistence of antibodies. So antibody indicates the presence of memory B cells. So all these systemic infections. What does systemic mean? It spreads throughout your body, typically via the blood. It can reach many organs and tissues. These all have very long persistence of antibody, 30 years, 75, 65, 40, 25, 40, 75. So this means there are B cells that last that long. But in contrast, mucosal infection, coronaviruses, flu, RSV, rotavirus, paltry, measured in terms of months. Measured in terms of months. So the question is, why? That's what Will says. Why? You know what? Sorry. We don't know. We have no idea. I once speculated and got yelled at. I don't know why systemic would have more robust long-term memory and mucosal would have lower. But obviously figuring that out would be nice, because if it's a simple thing, then you could build it into vaccines in some way. But it's a good question, Will. Don't know. And if anyone tells you the answer, they should say that they're speculating, okay? Because that's what they're doing. They are speculating because nobody knows the answer. Martha says, today biotech stocks fell on news of Peter Marks' departure. The strategy of not speaking up out of fear isn't working this time for companies to spell out the threat the upheaval at HHS poses. Boo RFK. Yeah, nobody wants to say anything. Nobody wants to say anything. It's really sad. Few people speak up. And, so virologists write something and they publish it in Journal of Virology. It's like, who are you writing for? Your colleagues, they already know the problem. You have to write for the public. Don't publish it there. Nobody's going to read it. It makes them feel like they're doing something. No, no, no. Mark wants to know, what microbiology will he find in Ireland? Similar to what you find everywhere else, but with local differences, right? They have microbes, you know that. Environmental, microbiomes, beer microbes. That's all I can tell you. All right, Steve says, saw YouTube with Doc Mike, surrounded by anti-vaxxers. You and Doc Offit should answer questions and refute misinformation to educate them. Doc Mike didn't have the extensive knowledge. Well, that's what we're trying to do on Beyond the Noise, right? Paul writes a column and we discuss it to try and educate people. It's 15 minutes. Try to keep it simple, but it's not. None of our stuff is really, really simple. And I agree that we have to do that more. John answers, Mark, perhaps some brewery microbes will be pervasive. You should let us know about their culture. Do we know how many viruses trigger autoimmunity? It's the best one, in my opinion, is Epstein-Barr virus. It triggers autoimmunity that leads to multiple sclerosis. But many others are suspected. Not proven, though. That's the problem. Not easy to prove. Like Coxsackie viruses and diabetes, thought to be an autoimmune reaction, but never proven. Never proven. So I don't give you a number. But there are multiple, and we don't know. Okay, this is a good question. How reliable is the information coming out of the CDC these days? I don't know how to answer that, right? I look at it, and it seems right to me. It seems like what they've been doing all along. They're not saying dumb things that are totally wrong, right? That you might think they might say. So I think it's okay so far, but it's only the beginning, right? These people are just getting their hands on this, so we have to be careful. Yeah. See you as a new president, yes. So, Bollinger was president, and then he stepped down, and then we had the lady from Egypt, I forgot her name, Mufa Sharak. I can't. So, and then she was followed by Katrina Armstrong, who was the dean of the medical school. So she took on two jobs, and she stepped down last week. And now we have Claire Shipman, who's a Columbia graduate. She's acting president. They still need to get a permanent president. That's going to take some time. And the administration has also hinted that she's not going to last long, because they found something she wrote in an email a couple of years ago, complaining about the usual Capitol Hill nonsense. And so one of the people on Capitol, I can't remember who it is, said, it just shows she doesn't take things seriously. To which I say, are you serious? Nothing the Republicans do show that they're serious. They just want to break things. This is the thing about them. They criticize you for what you do, yet they're above the fray, right? Yes, so Vanity, we have a new president. Hello, Jefferson, space in Miami. Welcome. Farhad says many ancient cultures recognize spring equinox, March 20, as a significant time for renewal and celebrations. Maybe that explains why April used to be the first month. I think it had to do with the Julian calendar. That's when it was set. But I don't know. What do I know about calendars? Helen from Pittsburgh, 16.7 C. 422 measles cases in Texas. Ah, yes, they're supposed to be public servants. I'm sure you heard about Cory Booker's um, uh, filibuster, right? And so it's one of the things he said, we're here to serve the people. We're not doing it. They're serving themselves. Enriching wealthy people. I don't understand why they have to be so mean. America's always been about taking care of people. And now all of a sudden it's not because a small fringe group of people think it should be another way? American voters, you better get your act together. Otherwise you're not going to have an America left. What about the law firms and the institutions of higher learning have done? What they have done is complying fascism in advance. I kind of agree. I don't like that they're throwing in the towel. Like UPenn for having a transgender person on the swim team 4 years ago. Really? You're going to punish someone for 4 years ago? So why don't we go back to 1960s and punish Columbia for the Mark Rudd demonstrations? This is ridiculous. It's just stupid. I can see last summer. Okay. It's very interesting, Carol. He couldn't buy Wisconsin, which is good, right? I think that's a really good. Unfortunately, we lost 2 more House seats in Florida. What are people thinking in Florida? Aren't they paying attention? Can't they see how screwed up it is and that they need it to go the other way? Never going to Florida in my life again. Sorry, you folks that live there. I'm not going, just out of principle. Okay, Mark Martin is bowing out of doing mini lectures because he says the TWIM did a good job. It's not the point, Mark. We want a different format, but I'll take care of it. I'll take care of it. Oh, he says he would love to. Okay, Mark, sorry about that. Maybe you could make a curated YouTube playlist of germ theory. One of the things I would like to do is have a chat with Carl Zimmer, who just wrote a book about this, right? That would be fun. Rach in the Isle of Man. It's going to be glorious tomorrow, 16C. I'm constantly saying it's unbelievable about every news story coming out of America at the moment. So now he's saying we're going to get Denmark, we're going to get Greenland, and I'm not ruling out force. What is this guy, out of his mind? I mean, this is just BS, but this is like what his voter base likes to hear. They want to go and conquer a country. This is totally contrary to the relationship we've had with Europe and many other countries. We respect each other's borders. We don't just invade for no reason, which is what he would do. He would invade for no reason just to, he would do it to distract what's going on here from what's going on. Just like Putin, I think, is doing this little war to distract from his screwing up of things in Russia. Trump thinks that's a good idea, but hopefully we can stop him. Right? He can't. I mean, many presidents have gone and initiated military operations before, but hopefully the one to conquer a country that's independent and has done nothing to us would be met with resistance. And if not, these people in Congress are just, I don't have a word for it anymore. Yeah, I'm missing Dixon too. I see his picture all the time, right? I left his name on all the schedules and notes. And I'm missing his chat with us. It's cool. Abdul Rahman from Manhattan, with the NIH cuts, can we set up some sort of public-private funded science communal bank of sorts? Yeah, we're talking about a lot of money here, okay? I am not... what am I not? I'm not opposed to doing it, but I think it would be hard to get the kinds of money, billions of dollars a year. The NIH budget is like $37 billion a year. And, a lot of people are saying, oh my gosh, that's too much. Yeah, really? You owe your health to that money. I mean, the return on investment for NIH is like 5 to 1. It does great stuff. Just take it away for a number of years, or misguide it, as is going to happen. Lots of very talented people who were head of institutes were fired this week also. And they're going to be replaced with people who are fringe scientists, pseudoscientists who haven't done anything good in their careers and made a career of just being contrary. Well, I put Jay Bhattacharya in that class. He should not be head of NIH, and I've explained why. But he is. Good thing I'm out of the grant game, because he'd look through the ranks and pull mine out and say, we're not funding Racaniello. Yes, Will points out that Adam Ratner, in his book Boosters, describes how measles in the Faroe Islands helped to confirm the germ theory when the miasma theory predominated. Yes, it was good. It was well done. Hey, MG, it was in the pathogenesis lecture. The longevity of antibodies of different viruses was in the adaptive immunity lecture. Spamlet says, RFK shutting down research that could prove long COVID wasn't caused by the vaccine. His law partner in Big Anti-Vax was on Mark Thompson shilling for him yesterday. Ignorance is money. Yeah, it sure is. It sure is. We have a lot of ignorance in this administration. I mean, this whole signal thing with the chat in the military operation. They look like such fools continuing to deny any wrongdoing because it's so obviously wrong. And now the jerk Mark Crow or whatever says on Fox, oh, I think Adam Goldberg, the editor-in-chief of Atlantic, I think he snuck in, putting this little grain of doubt in the Goldberg. I didn't sneak in. They just sent it to me. But they always have to put this shred of doubt in because people can't distinguish, right? Oh, my God. And the press secretary, the White House press secretary, they ask her a question. That issue is over, not answering questions anymore. How obnoxious, pure obnoxious. That's why she was selected, most likely. She could give obnoxious answers. Oh, my gosh. Yeah, so RFK is shutting down all kinds of research. You'll see, folks. You will see what he's doing. Yes, Dixon did like the Pat Metheny group, right? He did. Liked a lot of people, too. Okay, here's Chillin' Lillian. This was actually my hypothesis. I got yelled at for it. Maybe because virions flow through the blood and interact with different organs more in systemic infections instead of when the virions get stuck in the respiratory tract. So I thought, you got lymph nodes all over your body. So maybe they're interacting with a lot more lymph nodes than if they're stuck in the respiratory tract, and that somehow leads to better longevity of memory B cells. That was my idea, but I was told there's no evidence for it. No, there's no evidence. Just an observation. Yeah, we don't know why these systemic infections give long-term memory. I wouldn't tell you if I knew. I wouldn't tell you something if I didn't know it, and I don't know anything about it. So there you go. Okay, Aubrey, virology talk. Elon Musk went to Wisconsin, got his ass kicked by the woke mind virus. It took his money too. It's obscene that you think you can buy an election by giving people money. And you tried to change it, but one of the women he gave money to, she said, "Yeah, I did what he told me. I registered. I got people to register. Blah blah blah." It's just because he was yelled at by the judge. He said, "You can't do this. You cannot pay people to vote." I mean, these people don't know. They're so stupid. They have no common sense. Of course not. I can't expect them to have any common sense. Rainy is in eastern North Carolina, where it's 74F. Hans wants to know, do we know why only RNA viruses, such as RSV and rhino, and not DNA viruses, such as adeno and Boca, can cause infectious asthma? Is that true? They can, yeah, some DNA viruses can trigger asthma exacerbations. Sure, why not? I mean, it seems to be mainly RNA viruses, you're right, but I'm seeing adenoviruses here. So I don't have an answer for that because I don't think it's true. But RNA viruses are plentiful. There's a lot of them, so not surprising they're associated with asthma. Abdur says Doc Mike has basic knowledge and is more concerned with personal branding popularity. We shouldn't embolden more Prasads or Oz's. There's always a problem when people get in these prominent roles or quasi-internet prominent roles. They love the publicity. They love all the followers and the attention. Folks, I don't care. I just want to teach people. When I look at numbers, I'm just thinking of how many people we're teaching. I swear I do not care about branding and popularity. You have to believe me because that's what I'm saying. So we don't need someone who's interested in that crap. Thank you. Manoush Shafiq was the president before Katrina Armstrong. 3 presidents ago. Thank you very much. The AMA does a daily video on the latest stats. That's good. American Medical Association. I like that. I'll have to look that up. Boatloads lost jobs today in science. So horrible to send people emails at 5 a.m. And they say, it's not that you did anything wrong. You're just not aligned with what we want to do. Oh, wait, tell me what it is that you want to do. Elucidate it very clearly and comprehensively. They can't. They cannot. Going to shift attention to chronic diseases. This is the most BS answer. I bet it doesn't happen. Viruses cause a lot of chronic diseases. So you're just showing your ignorance. Hello, Barbara. Welcome to Office Hours. Dean wants to know how many microbe species are found in kefir and what is the mechanism that forms kefir grains. Well, Mark Martin, if you're here, please answer because I have no idea. No idea. Katya says, what about information on avian flu from CDC? I'm concerned we won't get real numbers. Well, you got CDC and you have the Department of Agriculture, so you could run them against each other. But actually, numbers are going down. The number of infections are going down. And guess what? Egg prices are dropping too. That's good. Natural events prevail sometimes. So I have, for example, the measles outbreak information at CDC seems reasonable so far. but what does reasonable mean, Vincent? Yeah, there's 400 cases. Maybe there are thousands and they're not reporting them. I just don't know. But the investigative reporters will find out eventually. They always do. That's great. That's why Trump hates them. The lunar calendar was adjusted because April was winter rather than spring. Okay. Yeah, Booker's filibuster was very good. I heard excerpts of it. He actually he was not scripted. He just talked, and he didn't read the encyclopedia that like Strom Thurmond used to do. Lise says, "Hello, Lise, from Ohio." Bollinger was president 21 years. Now the president lasts a year. It is a mess. I'm sorry, it was mishandled. Here's what I would have done. You can protest on campus. You cannot camp out on campus. Sorry. You protest during the day. At night, you're gone. She shouldn't have let them stay on campus. Then, if there's any anti-Semitic comments, death to Jews comments, out. 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 If you're free to protest, pro-Palestinian protest, but as soon as it gets violent, and violent means death too, that is violence. No camping, no violence. You can protest. That's what I would have done. And so at the end of the 5 p.m., goodbye. And that's it. Why the heck did they have to stay there? It's a mess. Yep. Farhad says live virus vaccines rarely cause unexpected issues, especially for immune suppressed people. Should we eventually fully switch to mRNA vaccines? No. No, we're not going to switch to mRNA vaccines. I think that we've had luck with a couple of attenuated vaccines. I mean, the MMR vaccine is perfectly safe. It doesn't revert. It doesn't cause the disease it's meant to prevent, right? Yellow fever vaccine, it's got some weird side effects, right? But it's a very good vaccine at preventing yellow fever. Infectious polio vaccine, OPV, nope, not good. Causes polio 1 in 1.5 million kids. Should never have been licensed. Sorry. So inactivated vaccine should be used for that. But we need next generation polio vaccines where not infectious, not inactivated, something else that hits your mucosa and gives you great immunity. And that's what is stopping polio research this summer. It's going to be very hard to do that. But no, I don't think mRNA vaccines are the future. Too many people are against them, including RFK Jr. They think they do bad things, which they don't. There's no evidence that they do any bad things at scale. They do some myocarditis, which is far less frequent than for infection and is treatable. And that's it. John wants to know, does the paralysis present the same from poliomyelitis, EV68, and other entros? Do they have distinct characteristics that only occur in particular viruses? Well, this is where I'd like to have Amy here. She knows this inside out. So, poliomyelitis is a distinct entity, right? Destruction of the gray matter and of the spinal cord and very specific parts of it, in fact. Other enteroviruses don't usually present as acute flaccid paralysis or myelitis. Now, EV68 can rarely cause a similar disease as polio, acute flaccid myelitis, but it has some different characteristics, and it's much rarer. Jill said she watched Cory Booker's speech for many hours. His speech plus the excellent folks in the YouTube chat really inspired me. Looking forward to Saturday's peaceful protests. Jill, I'm afraid someone's going to make them not so peaceful, okay? Someone from the admin is going to do something and they're going to blame the protesters, but it's going to be them that does something. I'm afraid for that. Vanity says Republicans won the 2 Florida seats, but they lost about half the advantage points that they had. Okay, I know, that's great. Maybe in 2 years they'll lose it all. Maybe enough pain in the next 2 years, that'll do it, right? Because I'm telling you, these treaties are not going to do anything good. Nothing. Peter says the 1951 treaty with Denmark let the U.S. set up bases wherever they want. Yeah, that may be so, but it didn't say they could just take over the country, right? Yvonne says, I got my MMR today. I guess I might be magnetic. That's right. Let's remember that. Margot says it's contrary to what America has stood for in my lifetime. It's the same way I feel, Margot. I grew up in this America where we helped people, right? I mean, we've done dicey things just like everybody else. But, we came to the help in Europe in World War II. We had to go in the Pacific region. And then over the years, we've helped people with medicines and so forth, which I think is better than war. And here in the U.S., we've helped our people, right, with Social Security and all these things that FDR put in place. And I was always very proud that it was a country that helped people. Why? Because it could. It could help people. The current administration says everybody's taking advantage of America. We're going to stop that. They're not taking advantage. First of all, they're human beings. They need our help. And I would give them as much help as they need. I've always said giving medicines is far better than giving wars. And yes, now we're reversing this, but I so hope that we can change it. It's not going to be easy, but I hope enough people realize what a crappy situation they've put us in. Oh, 2410, why is it difficult to create a fungal vaccine even though they're harmful? I think it's hard to identify the antigens, right? And no commercially available fungal vaccines. And you know what the problem is? There are antifungal drugs, but most of the infections occur in immunocompromised people. You can't vaccinate them. They won't respond well, right? But people are working on it. So maybe eventually you will see them. Can you do a series on fungi like you do for viruses? The reason I can do viruses is because that's my thing, right? I've studied them a long time. So it wouldn't be for me to do a fungal series. It would be somebody else. Quinn from Chicago, 65. I do TB, tuberculosis advocacy, and we are really sad about all this. Yeah. It's going to get worse. Tuj says, what are the chances of a fungal pandemic happening with global warming? Well, many people feel that fungi traditionally reproduce at lower temperatures, but as the climate changes, the fungi are adapting to higher changes, and they're going to get better at growing in us. And you won't have to be immunosuppressed. It will just grow in you because it can grow at 37 degrees. Not yet, but some people, Arturo Casadeval, for example, who think we're headed that way. Pamela writes, Columbia used to be the female personification of America before the Statue of Liberty became the new one. The uni should stand for liberty, too. She had a Phrygian liberty cap like the mushrooms. Is that who's sitting on the steps of Low Library? Is that, let's see, who is statue on Low Library? No, it's Alma Mater. I walk past that every day. Big, broad steps leading slowly up. Very nice. Yeah, this is funny. Norovirus outbreak on a cruise ship. Yes, Monday I taught about acute infections, and norovirus was one of them. I was talking about cruise ship outbreaks. Yeah. Hello, Quinn from Chicago. 65. Oh, I already read this. All of the national TB people got fired. That's new for the old one. Hello, Patricia from Massachusetts. Ted says he's been seeing several micro TV shorts recently. They're good teasers for introducing all of the This Weekend podcasts. We've been trying to make shorts, yeah. Yep. Yes, I heard the signal debacle is growing. Waltz had 20 additional groups going. Oh, no, I never do any confidential stuff. I'd have to lie outright. Oh, my God. Margot's going to start listening to Robert Evans. I have not heard of him. Parker, since me and my identical twin have the same DNA, doesn't that mean me and him would react to germs and vaccines the same? Well, actually, you don't have the same DNA. You were, maybe came from the same egg that split, right? But between the egg and you is many, many rounds of DNA replication, which is error prone. And so you have substantial difference from your twin. Let's see how much. How much does twin DNA differ? Yeah, this isn't a good answer. I'd have to look at a paper. They're saying there are 5 changes out of 3 billion. I don't believe it. There's no way I believe that. Anyway, you'll react somewhat differently, yes. Mike asks, is there an admissions filter to dissuade entryism at medical school? I don't know what that means. I looked it up, but I don't see how it looks in terms of medical school. But anyway, I don't know anything about admissions at Columbia, so I couldn't tell you. Angry Penguin writes, when POTUS says Canada should be the 51st state, does he realize the new state would have the same number of representatives as California and none of them would ever vote Republican? No, he doesn't, because he doesn't think. He just says stuff to make a splash, right? Without thinking what it means. That's with the tariffs. He just changes every day, and business people don't like that. Jill says they are using Gmail for other official business. Yeah, there will be no consequences for them. And in any other administration, they'd all be fired. Ridiculous. Gregory is in Bethlehem. 41F. Pollen season has begun. Viaduto is in Brazil. I discovered this channel a short time ago. How are things in Colombia? I recently discovered that you were a professor there. I'm not in Columbia, the country. I'm in Columbia University in New York. And so right now at Columbia, we have problems in New York City. So we read the newspapers. Just thinking if RFK came down with a good case of monkeypox, he would run. Of course he would. I don't think that he makes close contact with people such that he would get that. But it seems to me that he could get any number of infectious diseases, unless he's vaccinated up the wahoo and he's not telling anyone, right? I'm protected. I told you, he had a party during COVID. He required everyone get COVID vaccines to attend the party. Does anyone ever know about that? No. Nope. Patricia writes, when people are in the hospital, what symptoms do they have? Do you think if the doctors explain to the public what's happening, would that change anti-vaxxers? No. Because anti-vaxxers are listening to people that say things that they agree with. It's a Patricia writes later for measles, right? I don't know. I mean, during the Texas outbreak, there were a bunch of kids hospitalized, and RFK Jr. said, "Oh, they're hospitalized for quarantine." No, and they asked the hospital person. She said, "No, we don't quarantine for measles. That'd be ridiculous. You're exposing everyone else. We bring kids in the hospital with measles when they can't breathe. Stupidity." Tom says if they shift attention to chronic disease will be to tout various crank and fringe notions, sell useless supplements, etc. Absolutely. Totally the case. Totally the case. That's right. You have to make some good trouble. That's what Cory Booker said. John, who said that originally? John Lewis, right? He said you have to make good trouble. Make trouble, but make sure it's good. And that's what Cory Booker did. Yes. Polka Dot Pup asks, if the local small park lake has hundreds of large ducks, is it wise to not be down there tromping around in the bird poop? If they had bird flu, they'd be very sick. But, the poop on the lakeshore could be contaminated. So, I mean, you could look at them. I would stay away from the poop, right? Yeah, Gwen, Debunk the Funk, put out a really good video versus Joe Rogan and his guests. Yeah, he had this woman on, total anti-vaxxer. And so he emailed me earlier in the week to try and combat some of the crap she said about polio. She said in the 50s, the reason why polio vaccines worked is that it became something else, a different disease. It was no longer polio. Where do you get this crap from? You just make it up, pull it out of the air, and it makes no sense whatsoever. No sense. And she said doctors got fooled by a lot of other symptoms that looked like polio. No fooling. Polio is very clear. She's just full of misinformation. So I'm glad he did that. I sent him some answers. Hopefully it was useful. Baselta, nice to see you. Stay with us. Artemis germ theory. Louis Pasteur, Charles Chamberlain, among others. Koch in Germany. Semmelweis. Many people were involved, all contributing. Even microscope guy whose name I'm forgetting. First microscope, Leuwenhoek. He didn't really make the first one, but he made the first one that worked really well. Leuwenhoek. A lot of people were over the years feeding into that. Yeah. Hit the like button, folks, while you are there. We have 186 people. We have 120 likes. Please hit it. Help us to attract other people here. Brian Mack, we have a saying in Australia, it can only happen in America. It's true. We've had a lot of good things happen here. And now look at this. Okay, Baselta. What are your thoughts on the use of viral vectors for gene therapy, considering their emerging safety concerns? And using lipid nanoparticles. Well, lipid nanoparticles are just not going to persist forever. No, they don't. Not forever. Where these vectors can persist for your lifetime. And frankly, I don't think there are emerging safety concerns with them. There were with lentiviruses, with retrovirus vectors. That was fixed. AAV is a great vector. It's been licensed for a couple of therapies. So, no, I don't think LNPs are going to be the future of gene therapy. MN, thank you for your support of science communication. Really appreciate it. Did you ever see the YouTube video of the interview with the Paul Alexander, the last man with polio? I didn't see it, but I saw it when it was. I think he died recently, right? Or maybe not, but it was publicized recently. Sean wants to know if the flu is always DNA. No, it's RNA, not like us. As Mark says, minus RNA in 8 segments. Do you think CT scans cause cancer? I don't think. The data say it's the third biggest cause of lung cancer. Number 1, smoke. Number 2, radon. Basements. Number 3, CAT scans. Yes. So it's a tough one, right? Because it would be nice to do a CAT scan on people every couple of years. We do colonoscopies, right, to look for colon cancer, and that's great. It's not radioactive, but the CAT scan, it's radioactivity, and it would be sure good to pick up lung cancers at a small size, right? But that's not my field. But yes, third leading cause of lung cancers. Yep. Why is it hard for the immune system to fight fungal infections? It's not hard. If you're immunocompromised, it's hard. Most people can take care of them. Yeah. And yes, Daniel Griffin's Puscast does cover some fungal diseases. John, thank you for your support of science communication. My contribution is what I can do to help support Microbe TV. All the shows are exceptional and just a great perk to be commercial free too. Thank you, John. I appreciate those words. Lee says, look for the owl in the fold of the robe of the statue in front of Lo. I'll find it next time I teach. Take a picture of it. Yes. Eliza mentions epigenetics differ for twins too, right? So the histone-based regulation of transcription, that differs from a twin to a twin. Yeah. Here, Tractor Poodle pointing out previous anti-vax movements, 1879 and 1867. Mark George Herbie proposed high-risk CMV as a transforming agent in glioblastoma. I've not heard of that. I'll look into it. Thank you. Chillin says, can you talk a little more about where scientific papers on viruses should be published? In many places you can publish virus papers. So they're the virological journals like Journal of Virology, Virology, Viruses, and more. And then the luxury journals, Cell Science, Nature, Cell Host Microbe, Nature Communications, Nature Microbiology, plenty of places to publish. And that's what we do for TWIV. We look at all these sources and still find plenty of virus papers to talk about. Abdur says the return on investment on the NIH is actually amazing. If it was just for the Human Genome Project, it would have been worth every cent. It's true. It's really amazing. And these people don't recognize that at all, and they're just trashing it. Hello, Bad Cat. Welcome from Maine. Hello, Kip. Thanks for your support of science communication. I'm strong. I'm doing my best. I'm keeping it up. Not getting frustrated. I'm a little sad about what's happening to this country, and that certain people can't see what they're doing. They just tell you, oh, we're going to make it better. Really? So what's the exact plan? Trust me. Nah, sorry. I don't trust. I want to see the plan. Abdur Rahman asked if a mother with an active chickenpox infection gave birth, should she be separated from the baby? I don't know. That's a good question. Always a good question from you guys. Yeah, there are a lot of problems that can happen. They don't mention separating the baby, but they do mention treating the baby with antibodies, right, and antivirals to prevent spread of infection. Good question. Mike says entryism is someone getting an MD but for another agenda, like pushing homeopathy. Well, I've seen medical students get MDs and they want to do other things like IT. They don't want to treat patients. They just want to do IT and want a medical training. And, that's hard because we do need doctors in many parts of this country. But a lot of people don't want to go to those places, right? So I've interviewed people, medical student applicants, who want to do other things. Yeah. Can Fauci sue RFK for his book? Yeah, it's quite slanderous, but I don't think he's going to do it. He doesn't want the notoriety. It's pretty much all lies, right? He says Tony Fauci is the worst human being on the planet. He doesn't care about anything. This is just slander, right? It's lies. And other things too, of course. Okay, let's do a mini lecture, and then I'll come back to questions. So this is oh, I got to take Will off there. Yeah. Okay. This is part of a lecture that I give in my course called The Infected Cell. And what is that about? It comes at the end of the first 10 lectures where we talk about reproduction of viruses in cells in culture in the lab. And now we're going to talk about, before we go into disease, this is an overview. Basically, we talk about viral gene expression, genome replication, assembly of virus particles. All of this depends on the host very much, right? Metabolic pathways, biosynthetic pathways, signaling, and trafficking systems. So this lecture is meant to be an integrated description of what virus infection does on the host cell in terms of metabolism, biosynthesis, signaling, and trafficking. And I'm not going to go through all of those tonight. It's too much. It's overwhelming. I'm going to keep it simple. So normally we would talk about signal transduction, gene expression, metabolism, and remodeling of cellular organelles. And I'm just going to talk about the last two, metabolism and remodeling of cellular organelles. Now, the thing here is that when a cell's infected, making virus particles you need precursors. It places high demands on host cell biosynthetic systems. You can't just go in and make a lot of viruses. You have to turn things up. You need to make nucleotides. You need to make amino acids. Nucleotides for genomes, amino acids for proteins, fatty acids for membranes. And above all, you need energy. Look at this. ATP is one form of energy, right? You hydrolyze it to ADP, you get some energy. 4 ATP are needed to make a single peptide bond. What is a peptide bond? It's a bond between 2 amino acids and a protein. So you can have a protein 1,273 amino acids long. Does anyone know what protein is that long? It's the spike protein of SARS-CoV-2. You multiply that times 4 ATP, except for the ends, I guess, to figure out how much energy you need to make one spike protein. It's enormous. So what happens when a virus infects a cell? It turns up metabolism. It turns up the thermostat. And that's what I want to talk about a little bit. Here's the Krebs cycle, the tricarboxylic acid cycle, which is a central hub of carbon metabolism in cells and generates lots of energy in the form of GTP and NADH, etc. We'll get to that. But first, look, very simple experiment. You infect cells with adenovirus. And we infect cells in these little dishes. So these are plastic dishes. On the bottom of them is a sheet of cells. And they're living, and we have this medium in them, which has a pH indicator in it. So if the medium gets acidic, it'll turn yellow. Okay, so we infect these cells with adenovirus. First one is mock. We just put saline on the cells and go through the whole process. 24, 36, 48, 72 hours, you can see. No change in the pH. Adenovirus. Look, it's turning yellow, 48 and 72 hours. The pH is dropping. Why? The production of lactic acid, which is a product of glycolysis, the breakdown of glucose. I'll show you that in a bit. And here's an adenovirus, a mutant that doesn't replicate. And you see it doesn't make the medium acidic. Okay, so that's a nice visual experiment. But on the right, we're actually measuring glucose consumption, nanomole per million cells per hour here on the y-axis. And then we have these experiments here, mock 24, 48, and 72, wild type and our mutant. And you can see the wild type consumes glucose substantially by 72 hours, which we knew because the medium was yellow, right? And the mutant does not. Okay, so many viruses do this. Rhinoviruses have a sweet tooth. They need glucose as well. Okay, why? Why do they need glucose? Glucose is the major breakdown product of dietary carbohydrate. You eat carbs, it's broken down to glucose. And then the glucose is taken up into the cell. Here at the top, here's a cell. The membrane of the cell, plasma membrane, is shown at the top. And glucose is actively brought into a cell by a transporter. Glucose transporter, GLUT. Here it is now in the cytosol. And then it undergoes a series of biochemical reactions, many of which generate energy. And you need a little energy to begin with to get things going. So you need 2 ATP. And then you get 4 ATP made in the end. But then there are other. So we take glucose, we make glucose 6-phosphate, fructose 1,6 bisphosphate, and other compounds. And many are not shown here. Pyruvate kinase makes pyruvate, which goes to acetyl-CoA, the precursor for fatty acids, and then eventually lactate. Pyruvate goes to lactic acid, which makes the medium yellow. Isn't that cool? Now, you can use acetyl-CoA to make membranes. We'll look at how that happens later. And you can also take glucose 6-phosphate way near the beginning and shunt it off to what's called the pentose phosphate pathway, and that makes ribose 5-phosphate, which is the sugar in nucleic acids, right? That ribose part of each base, ATP, has ribose in it. This is where it comes from. So, wow, the things you can do with glucose. You can make energy, you can make nucleotides, and you can make fatty acids. Now, if you have too much of it, it's stored as either carbohydrate like glycogen or fat. Anyway, this is perturbed in virus-infected cells, right? Because viruses need energy, they need fatty acids. So let me show you one way that it's perturbed. So here we have two viruses, herpes simplex virus and human cytomegaloviruses. These are two herpes viruses that have different effects on glycolysis. This is, again, the glycolytic pathway. We're just kind of abbreviating it here, right? So for human cytomegalovirus, it actively transports glucose into the cell. That's the green arrow. Glucose 6-phosphate, pyruvate, goes all the way to acetyl-CoA, which is the precursor for fatty acid synthesis. So, okay, that's cool. HCMV needs membranes. But herpes simplex virus does something different. It doesn't actually it doesn't stimulate the uptake of glucose, but what it does do is it shunts it off early on in this pentose phosphate pathway to make nucleotides, to make ribose 5-phosphate and nucleotides. We don't get pyruvate in herpes simplex virus infected cells. So different effects of two herpes viruses on glycolysis, the breakdown of glucose. So the idea is that the herpes simplex viruses have shorter reproduction cycles, and they need more nucleotide precursors. They need to make a lot of DNA, whereas HCMV is longer, and it's okay dealing with the nucleotides that are available, but it needs a lot of fatty acids for membranes. So that's just an example of metabolic effect of viruses on cells. It's really quite substantial, and this is not something we knew 15 years ago. We just learned about it. And the reason we did is because we figured out ways to study metabolites in virus-infected cells. Okay, fatty acid synthesis, that's another one. You need acetyl-CoA to make fatty acids, all right? But here's the mitochondria right here, right? And this would be the cytosol of the cell. Acetyl-CoA cannot cross the mitochondrial membrane. And so what happens is pyruvate is imported into the mitochondria, and then in the mitochondria it is converted to acetyl-CoA and then citrate, and then it's exported as citrate, and then that goes towards the synthesis of fatty acids. All right, so we have citrate going to oxaloacetate in acetyl-CoA, which is a reversal of the first step in the TCA cycle, and that needs energy. And then the second step, you convert acetyl-CoA to malonyl-CoA, also needs energy. And then the malonyl-CoA is converted to fatty acids by fatty acid synthase. All right. So this reaction has to occur in the mitochondrial matrix, but acetyl-CoA can't get out, so that's why it's converted to citrate. Now, if you inhibit malonyl-CoA or fatty acid synthase, you reduce the yield of human cytomegalovirus because it needs lots of lipids for its membranes. And any virus with an envelope is going to modulate this cycle. Like VSV, the activity of another member of this shuttle, which is not shown here, is increased in cells infected with VSV. Okay, fatty acids. So the output here is fatty acids. We'll come back to those later. Now, the citric acid cycle, or the tricarboxylic acid cycle, you probably learned this back in high school and college and then promptly forgot it. But it's really important. It's the central hub of carbon metabolism. It yields precursors for the biosynthesis of many compounds, including pyrimidines and purines, fatty acids, and other things. And essentially what happens here is that we have pyruvate from glycolysis converted to acetyl-CoA, and acetyl-CoA gives 2 carbons to oxaloacetate and becomes citrate. And then it starts to be converted to other compounds. And along the way, some energy can be made right there, NADH, NADH, GTP, NADH. So this conversion is generating energy. Now, virus infections impact this cycle. For example, herpes simplex virus needs precursors to nucleotides. So it converts oxaloacetate to aspartate, which can then be converted to pyrimidines and purines for the synthesis of NTPs. However, if it kept pulling oxaloacetate out of the cycle would stop, right? Because there would be no oxaloacetate to go to the next compound. So what herpes simplex virus does, it inputs glutamine. It converts glutamine to alpha-ketoglutarate down here, which replenishes the cycle and prevents it from halting. How thoughtful these viruses are. And human cytomegalovirus needs fatty acids, as we already showed. So it pulls citrate out of the Krebs cycle to make fatty acids, and it inputs glutamine to prevent halting of the cycle. So just some examples of how virus infections impact the TCA cycle. Really cool. Okay, a little bit on lipid metabolism. So this is a lipid. It's a triacylglycerol. It has fatty acids and a glycerol backbone, okay? These triacylglycerols, there's 3 fatty acids, are the primary energy source and energy store in most organisms. This can be oxidized to make energy, and it can be stored. And how it's stored is shown here on the bottom. So these lipids circulate in the body associated with proteins called lipoproteins. And to store or to use the fatty acid, the lipoprotein is acted upon by lipoprotein lipase, which makes free fatty acids. So that cleaves the 3 fatty acids from the glycerol, and the free fatty acids are imported into the cell. Acyl-CoA synthase makes acyl-CoA from them, not acetyl, but acyl-CoA. And this goes through the ER and eventually coalesces into a lipid droplet. So many lipids coalesce into this lipid droplet, which then has a protein coat. So this is called a mature lipid droplet, and it's a store of lipids inside of a protein coat. Now, viruses need to modulate the balance between synthesis and oxidation of fatty acids because they need lipids for membrane synthesis, right? And so lipid metabolism is modulated in virus-infected cells to balance or to regulate oxidation versus synthesis. So I'm going to show you this. It's a little complicated, but let me try, okay? Now, human cytomegalovirus is unique. It needs very long-chain fatty acids for assembly. What is a long chain? So this is the fatty acid. These can get longer and longer and longer, okay? They're of different lengths. So cytomegalovirus needs very long-chain fatty acids for assembly. So this is why the virus infection increases carbon flux, the movement of carbon from glucose to acetyl-CoA. Remember, I showed you that. Let's go back. I'll show you again, right? So HCMB moves glucose all the way to acetyl-CoA. No shunting off. Okay? And this results in increased fatty acid production, but also the long-chain fatty acids that the virus has in its membrane. The way it does it is shown on the left. All right, so the genes to make fatty acids and sterols are encoded, of course, in DNA. And they are driven by a promoter. And in particular, there's a protein called SREBP1M, which drives the transcription of these genes so that you can make fatty acids. All right? So how does human cytomegalovirus turn this up? Well, this transcription factor is normally not in the nucleus. It's normally in the endoplasmic reticulum. It's right here. It's hung up because cells don't need to make fatty acids all the time. But cytomegalovirus infection causes the release of this transcription factor from the ER. It goes through the Golgi. You can see there's this transmembrane part. The actual transcription factor is cleaved off, and it goes in the nucleus. And there is a protein in cells that blocks import of this protein in the nucleus, and human cytomegalovirus antagonizes that so that the protein can get in, and it can turn on the genes for fatty acid synthesis. How cool is that? So the virus perturbs the ER, so that releases SREBP1P, and it becomes released eventually from the Golgi and goes in the nucleus. That's how HCMV turns on lipid synthesis, in particular long-chain fatty acid synthesis. Okay, same thing with other viruses. Hepatitis C virus is an envelope virus. It needs lipids. And not only for the membrane of the virus, but it makes vesicles on which to make RNA, on which it makes RNA. So here's an experiment where we have mock-infected cells and HCV-infected cells. These are human hepatocytes. The target for HCV is the liver cell, the hepatocyte. These are stained with a dye that stains lipids, so green. So mock-infected cells, there's a basal activity of lipid synthesis. But virus infection, look at it, turns it way up, kind of like HCMV turns it up, but it's a different mechanism. In the case of HCV, I'm going to show you how oxidation and synthesis are balanced, right? So, the outcome of lipid metabolism is a balance between lipogenesis, making lipids, and fatty acid oxidation. And, these are each controlled by genes that are driven by specific transcription factors like the one for HCMV, FOXO1 and FOXA2. So FOXA2 drives oxidation and FOXO1 drives lipogenesis. So when HCV infects a cell, the cell has FOXO1 stimulating the production of transcription proteins that induce lipogenesis. In fact, here's our friend, SREBP. And one of the genes that this transcription factor will induce in the nucleus is fatty acid synthase, which will give rise to new lipids. So FOXO1 doesn't have to be in the nucleus all the time. So it goes out until it's needed. But HCV infection blocks its exit from the nucleus, so it will stay there and keep making these proteins needed for lipogenesis. All right. If you knock down the levels of FOXO1, you inhibit HCV replication. So, on the one hand, HCV stimulates lipogenesis by retaining this transcription factor in the nucleus. What about oxidation? The enzymes that oxidize fatty acids are shown here, MCAD and SCAD5. These are all under the control of another transcription factor called FOXA2. And when FOXA2 is present, these genes will be transcribed, the proteins will be made, and you'll get fatty acid oxidation to get energy. But what Hep C does is it downregulates the production of this transcription factor so that you reduce oxidation of fatty acids, because the virus requires them for reproduction. And again, if you overproduce FOXA2 in a Hep C infected cell, you inhibit replication because you're driving fatty acid oxidation. And so that's how Hep C manipulates the fatty acid program, both synthesis and oxidation. Now, many viruses remodel cellular organelles to make sites for reproducing their RNA. And here's an experiment with polio. Just like the one we showed before, polio mock-infected cells stained with a dye that stains lipids. Polio infection increases the synthesis of lipids massively. And what happens is the viral proteins actually recruit lipid droplets that are the stores of lipids in cells and they take away the fatty acids, make phospholipids, and make these membranes on which to reproduce their genomes. And that's shown here in a poliovirus infected cell. The ER and the Golgi are gone, and they're reassembled into these vesicles on which viral RNA can be synthesized, and that's why lipid synthesis goes up in virus-infected cells. Same thing happens with hepatitis C virus. Hep C assembles on membranes. This is an ER membrane here, and these are vesicles that have come off it. And these are all viral proteins and RNA in here. And eventually a lipid droplet gets assembled in here, and that serves as the assembly point for viral particles, which you can see then bud into the endoplasmic reticulum. So all of this is membrane taken from the host cell. SARS-CoV-2 does the same thing, right? It infects the cell, generates these double membrane vesicles from lipids present in the cell, and the genome replicates on them and eventually gives rise to new virus particles. But this is a cryo-EM of these double membrane vesicles in which all RNA synthesis occurs. All right, one last bit of information which I think is fascinating. This is actually a plant virus, tomato bushy stunt virus, which infects tomatoes and makes them small so you can't use them. This virus is an RNA virus. It also reproduces in its RNA in vesicles, but it does so on the inside of the vesicle. And so that's what's happening here. Viral RNA synthesis is happening in the vesicle. Now, you need energy for this to happen, viral RNA synthesis. And so what happens is the viral protein p33 grabs pyruvate kinase and brings it into the vesicle so that it can make ATP. So pyruvate kinase converts ADP to ATP. And that's brought in by this viral protein p33. So here's a biochemical experiment that shows that. We have cells, these are infected cells, and this one is stained for p33. So that's the protein that's brought into these vesicles. This one is stained for pyruvate kinase. So now you can see this little vesicle, which is diagrammed at the right. It's full of pyruvate kinase, which is the enzyme that's brought in to make energy. And then if you merge, you see that the p33 and the pyruvate kinase are in the same vesicle. And so that's the evidence that p33 brings in the kinase to make ATP, which is needed for unwinding double-stranded RNA. Isn't that cool? That things you can learn when you do research. It's amazing. And why you'd want to break this, it's because you're ignorant of the knowledge that's been generated. Okay. Let's get back to our questions. Eliza is reading The Song of the Cell by Mukherjee. He describes Leeuwenhoek's microscope. I haven't read it. I read The Emperor of All Maladies, right? And then his next book had something to do with DNA. People said he totally messed it up, so I didn't read it, and I haven't read any one since. Look at this. Mike says the Conservative government in Ontario has demolished the Science Centre and will downsize it. Conservatives are anti-science. I don't get it. They benefit from it. Paul Alexander died in 2024. Yeah, I remember that. The last polio survivor or a polio survivor. I remember that very much. Yeah. Venture, Ontario. If you were streaming tomorrow, it's payday. I would donate it. I've been watching you since before COVID. Unfortunately, my mother loves RFK Jr. I don't know how you could love a guy that wants to harm kids. Maybe you should point that out to her. Ann is checking in from Tucson. Hello. Has RFK banned any vaccines yet? No, not outright. But, he didn't go to Texas and say you need to get vaccinated and push measles vaccine. No. So kind of a soft negating of it. But just keep watching. I'm sure he'll ban something. And we'll talk about it. Mike says, don't these politicians take vaccines for travel? Of course they do. They're all fully vaccinated. They're liars. Vaccines work. Ay. I think this administration is trashing many of the most accomplished people and fascists to terrorize us and to steal the dough for themselves. Yeah, totally true. I agree. Visto asks, when viruses bring their own polymerase into the cytoplasm, is it different from human? Yeah, it is different enough to make inhibitors, yeah. But I'm not aware of any that have been licensed. But yes, it is a target. Hello, Eleanor is in Toronto. Freezing rain. I have diagnosed with spinal cancer in 2019. Have a CT scan every 6 months. I think it's a long-term effect, CT, on lung cancer. I don't think you get it after a year or two. It's probably like 20 to 30 years, right? But yeah, it's always a trade-off with cancers, right? You trade off one thing for something else. You wouldn't get a CT scan for your brain, I think. Well, I'm not a doctor, I don't know, but maybe you could, but the MRI would be much better to see what's going on in your brain. Leonard saw a couple of papers that the EV68 proteinaceous receptor has been identified. Yes, we did it on TWIV. It turns out it's not a very good paper. And multiple papers have attempted to identify an EVD68 paper, and I think all of them fall short, unfortunately. So we simply don't know yet. Eleanor, thank you for your support of science communication. Appreciate it a lot. We need everyone's support, and I really am very grateful when you come to these office hours and support us. Chillin Lillian, wow, the consumption of glucose is fascinating. It's such complex behavior for siphoning resources, all done passively. That's right. It's all passive. Virus gets in, makes proteins, they have an effect. There's no intent at all. There's a connection, Abdul writes, connection between CMV and glioblastoma. Seems the tumor can predispose to reactivation of the virus. Yes, so we all have latent CMV in us, and then the tumor reactivates it, and the virus helps make the tumor more aggressive. Interesting. Interesting. Hoping for CMV and Epstein-Barr virus vaccine soon. Yeah, we're working on them. They are working on them. Although, it's academic research. Is it going to continue? Yeah, Mark makes a good point of it. Representative who only cares about money. Vaccines are so much cheaper than treatments. There you go. But nothing they do is logical. Mark, you know that. Yes, this is it, Vanity. The Krebs cycle. Memorize, forget, memorize, forget, memorize, forget. Well, my students don't have to memorize it because they have open book exams. And so now it makes perfect sense in terms of virology. Pete met Krebs' son at an AGM of the London Cycling Campaign a number of years back. Useless factoid of the day. That's interesting. I like that. Vanity met one of his relatives on a flight. Huh. Krebs. His cycle. Does CMV and herpes differ because they're targeted? No, I think it's all about how quickly the reproduction cycles have to go. Richard from Northern California, or Dairyland, heart of the H5N1 pandemic. Welcome, Richard. Very Nika says, I wonder how all this influences the host. Does the person even feel that their energy is stolen? I think on a host level, it's very small because you continue to eat and nourish your cells. You do, for many viral infections, you do feel malaise, but that's more an effect of immune responses than increases in metabolism. Yeah. Oh, Nevitz helped crystallize SREBP protein bound to DNA 25 to 30 years ago at Rockefeller. It's still a big deal, Nevitz. Good job. That's awesome. Will the virus continue depleting cell resources until it collapses? Yes. In the end, it uses everything up and you can't make any more viruses. Yeah. Well, welcome, Pen Click. It's just me, but it is live, yeah. Humans and viruses aren't that different. We both come into an environment and immediately start consuming all the resources for our own gain without any thought for the future or who it impacts. It's true, Chillin. It really is true. We're the worst enemies for Earth. You're absolutely right. Because we're very selfish. We just want to survive. Now, just sitting there slurping up all that poor tomato's energy. It's right. That's right. Yeah, I mean, viruses are the product of many, many years of evolution. So they do it right, Yep. Okay, now we're talking about the Matrix. So Abdur says, someone saw the Matrix, Chillin Lillian, I guess what she just said about consuming all the resources. And then very neat is, oh man, I remember watching The Matrix the first time as a nihilist teenager, and the Smith speech absolutely blew my mind. Bad Cat says it's too complicated for evolution. It must be intelligent design. Yeah, this is nothing that a human could do, or superhuman. It's definitely evolution. In a recent video, a situation was mentioned about a child who passed as a direct consequence. All right, so I think you're saying USAID. So USAID provided money for people in other countries to treat their HIV infections. So this child had been born to an HIV-infected mother. I think this is what you're talking about. Had been born to an HIV-infected mother. He contracted AIDS. But the U.S. supplied drugs to keep him alive. And he was, I don't know, 5 or 8 years old. And then the U.S. canceled USAID. Musk just turned the switch off. He didn't get his drugs. HIV started reproducing, and he got pneumonia and died. A direct effect of us. And Musk has said, no, people have died from turning off this money. That's a lie. That's a friggin' lie. Because this is, and there are others. There's a whole list of kids who have died, very personal stories, because we could no longer supply money for their medicines. This is not something I am proud of. It's horrible. I don't even want to travel anymore because I don't want people to look at me and say, what's with you? But it's not me, because I don't want this to happen. And Nevitt says it's worth repeating. All of these intricate pathways took hundreds of research scientists and technicians, decades of work to figure out. And there's plenty left to learn. Yep. Here's the same question. Is there a clinical impact on the host? I'm not aware that these energy demands have a clinical impact. It's more of the immune response that does, yeah. But of course, if you exhaust the resources of a cell and it dies, that's going to have a clinical impact, right? If it's a neuron infected with poliovirus, you're going to get paralysis. If it's some other kind of cell. So I guess, yes, if the end point is cell death caused by exhaustion of resources, then yes, there is an impact. Nika says, frankly, very annoying that our cells know how to perform the Krebs cycle while we struggle to understand it, let alone remember it. Yep. Isn't that cool? It's all working, but well, first of all, we don't know a lot of it, but what we do, it's hard to remember. But if you used it every day, if you thought about it every day, you'd remember it. But most of us don't. Yeah. So Hamlet asks, has anyone done a history of how all the organelles, biochemicals, and metabolic pathways were discovered and mapped out? That's a good question. There's certainly lots of histories of molecular biology, but biochemistry, I don't know. That would be some book, right? That would be very interesting. I don't know. I'm going to look it up. Maybe someone else knows. That's great. Justin, began watching your lectures. Can you make a video about lat expression in herpes? I read a paper. It determines the length of latency. I don't know about that, but I'm happy to do it. Did you see astronauts have continuous outbreaks of herpes? I didn't know that. Because they're under stress conditions? I don't think you should have to memorize the Krebs cycle. I think you have a picture of it. You understand what's going on, And you can look at it and get that. That's all you need to know. Yeah. Sorry, you just missed the mini lecture. Yeah, watch it tomorrow. Yeah, PEPFAR is the anti-AIDS program. That's right. I'm sure that some USAID money goes to it as well. Viruses are obligate. Maybe humans are worse because we have agency. Maybe. It's a good point. But Visto says humans are obligate on the planet Earth. We just don't always know it. That's really good. I like that. Mike, how do I arrange to visit the incubator? Send me an email, vincent@microbe.tv. Yeah, Nick Lane wrote all about the TCA. That's right. And other biochemistry as well. It's very good. Do viral infections have an effect on diabetes? Well, some people think diabetes is an autoimmune reaction to some virus infections. But I know what you're asking because of the metabolic things we've talked about today, but I'm not aware of any. No. Triad wants to know if coronaviruses increase the use of zinc. Let's look it up. I don't remember. I always forget. Do corona require zinc? No. All I'm getting are crappy articles on using zinc in COVID, but many viruses require zinc, so I would be surprised if coronaviruses do as well. John wants to know if anyone has tattooed the Krebs. I'm sure. Absolutely. Everything else in science has been tattooed. I once saw a tattoo, and I figured out it was a compound. It was some hallucinogen. I had to look it up, right? So I copied the structure and put it in Google, but she had this hallucinogen tattooed on her. Vista wants a mini lecture on herpes. Yeah, we can do that. I gave the full lecture today, but we can do that. Hello, Sabrina. Welcome. Unlike love, herpes is forever. If you hear that once, you never forget it, right, Vanity? They shouldn't pronounce USAID because it sounds like handouts. USA International Development. Yes. Yes, I agree. And then Les says USAID funds PEPFAR. Thank you. Is it possible for HSV1 to cause chronic migraines? Well, there's been a couple of studies showing an association between herpes simplex infection and migraine. We know that it does go in the brain quite regularly, so yeah, it's good. Not just theoretical, but there's an association, all right? So that's not proof, but it's an association. Really, Questella? You want to cover yourself in tattoos? Okay, whatever you want to do. It's your body. But science tattoos are very cool. Viruses, metabolic pathways, molecules. I think it's cool. Aubrey wants to know What is the measure of viral load and its relationship to the virulence of a pathogen? Measurement of viral load should be an effectivity assay, like a plaque assay. And viral load is a reflection of fitness. So the more fit a virus is, the higher the viral load when it's reproducing in cells or tissues. And often, higher viral loads are associated with greater pathogenesis. And so sometimes the selection for increased fitness will end up causing more pathogenesis indirectly. Okay. Or does it matter if viruses need zinc or not? Well, if they require zinc, then we could remove it as an antiviral approach. That's one reason. But also it helps to understand the pathways, and that may identify other targets for intervention. Hello, James from Austin. How's it going down there? Tractor Poodle says, when I was an undergraduate biochem major in the old days, we had to memorize all the pathways. I made a C but an A in every biophysics course. Principles rule. Yeah, principle is much easier. Okay, now we're talking about tattoos. If I were to cover myself with tattoos, it would be buckyballs and virions. That's cool. And Renata says, almost everyone I know who's covered in tattoos dated a tattooist. Really? Yeah, Will, this is true. Destruction of USAID and PEPFAR means the USA is abandoning soft power. So this will increase respect for USA worldwide. No, he doesn't care about that. He thinks that countries are taking advantage of the US by taking this money. Okay? But it doesn't work that way. They're not taking advantage of us. We are able to help them. All right? We are able to help them, and so we should. We have a moral obligation, but they have no morals. But other countries will step in to take over, I'm sure, because there's plenty of money out there to do this. Yeah. All right, folks. Time to wrap up this. Thank you all for coming tonight. And we have more likes than users. Some people left. I appreciate it. Let me thank our moderators, Vanity Nutrition, Les Fabby. I know Steph was here. Tom Steinberg was here. And who else? Did I get everyone? I got Vanity, Steph, Tom, Les. I'm missing somebody. Steph, Vanity, Les, Tom. There's no Barb Mac UK. There's no Frank. Okay, I guess that's it. Thank you for moderating. Really appreciate it. Thank you all for coming. Really appreciate your questions. Oh, thank you, Iron Felix. Been a listener for years, never thanked you live. So thank you. Thank you for coming, Iron Felix. Really appreciate that. I will be back next week. What's the date? Today is the 2nd, so next week must be the 9th, right? There you go. The 9th of April. The days are going by very quickly, aren't they? Okay, folks. Thanks for coming. I love the chat. I love your questions. It's all good. I hope you learned things. I hope you get stuff off your chest. And come back next week with more questions. In the meantime, I want you to be safe until next time. Thank you, everybody. Good night.